Hello students, today we are studying biology class 11th, chapter number 7, cell division. We have already started this chapter in our last lectures. We have studied the chapters, okay. Today we will continue with the chapter, okay. In our last lecture, what we have studied? We have studied cell cycle, types of cell division, mitosis, stages of mitosis, significance of mitosis, and then we started with the process of meiosis. Okay, let me revise the definition of meiosis. The form of cell division by which gametes with half the number of chromosomes are produced is known as meiosis. In meiosis, diploid number of chromosomes are getting reduced to haploid number. Meiosis is sexual reproduction. The term meiosis is coined by J.B. Farmer in the year 1905. The cells in which meiosis occurs are called as meiocytes. We have already done with this part. Okay, I have just revised the definition part of meiosis. And then we have started with phases of meiosis 1. Meiosis are of two types, that is meiosis 1 and meiosis 2. We have started with meiosis 1. Meiosis 1 consists of 4 phases, that is prophase 1, metaphase 1, anaphase 1, telophase 1. In that, we started with prophase 1 and we have completed the sub-stages of prophase 1, that is leptotene, zygotene, pachytene, diplotene and diakinesis. So, we have already completed till here. Today, we will continue with the next phase, that is metaphase 1. Prophase 1 we have studied. Let me revise it in short, that it is the longest and the most complex phase. Chromosomes are condensed in this phase. Synapsis occurs, that is homologous chromosomes come together to form a tetrad. And tetrad means 2 chromosomes or 4 chromatids are coming together. It is known as tetrad. Okay, so we have studied this prophase 1 in detail in our last lecture. That is all the substages: leptotene, zygotene, pachytene, diplotene and diakinesis. Let us continue with metaphase 1. What is metaphase 1? In this, all the bivalents migrate within a cell and migrate to the metaphase plate, that is at the equator. You can see here, this is metaphase plate and all the chromosomes come together and they come at the equatorial plane. One homolog is pulled above the metaphase plate and the other below. The centromere of the homologous chromosomes of each bivalent stretch out on either sides. So the centrioles are at the opposite poles of the cell. Spindle fibers from one pole of the cell attach to the one chromosome and spindle fibers from the opposite pole attach to the homologous chromosome. It is very simple. I will again explain this metaphase in short. The spindle fibers become well developed here in the metaphase 1. The tetrads move towards the equator. You can see here the tetrads are all situated at the equator. The metaphase means middle. All the chromosomes are coming in the middle, that is at the equatorial plane. The tetrads move towards the equator and they orient themselves on the equator in such a way that centromeres of homologs tetrads lie towards the poles and arm towards the equator. Due to increasing repulsive forces between homologous chromosomes, they are ready to separate from each other. So that separation we will see in the next phase, that is known as anaphase 1. So let us study anaphase 1. In anaphase 1, chromosomes move to the opposite poles. Okay, chromosomes moves to the opposite poles. The microtubules and the kinetochore fibers interact, which causes the movement. A difference between the mitosis and meiosis is that sister chromatids remain joined after metaphase in meiosis 1, whereas in mitosis they separate. During anaphase 1, original chromosomes separate, so reduction in the number of chromosomes from 2n to n number, yet the sister chromatids remain together. I repeat, it is also very simple phase. What is happening in this phase? Homologous chromosomes are pulled away from each other. See, you can see here the homologous chromosomes were at the equator, and in this stage what is happening? These homologous chromosomes are pulled away from each other, okay, and carried towards the opposite poles. These are the two poles. Chromosomes are moving away from each other and they are moving towards the poles by spindle apparatus. This process, when chromosomes are moving away from each other, it is known as disjunction. The two sister chromatids of each chromosome do not separate in meiosis 1. Okay. So, let us move further. This is reductional division. Okay, this is known as reductional division. The sister chromatids of each chromosome are connected by a common centromere. Both sister chromatids of each chromosome are now different in terms of genetic content, as one of them has undergone the recombination. So, what is happening in anaphase 1? Basic thing that you have to remember, that the chromosomes are moving away. You can see here chromosomes are moving away from each other. They are moving towards the poles and with the help of spindle fibers. This process, when chromosomes are moving away from each other, are known as disjunction. Okay, this process is known as disjunction. Let us move further. That is telophase 1. Next phase is telophase 1. What happens in telophase 1? The homologous chromosome complete their migration to the two poles because shortening of the spindles. Okay, the nuclear envelope organized around two groups of chromosomes. The nucleolus also reappears. Okay, what is happening here? The nucleolus reappears. I repeat, the haploid number of chromosomes after reaching their respective poles becomes uncoiled and elongated. You can see the chromosomes have become uncoiled and elongated. The nuclear membrane and nucleolus reappears. This nuclear membrane and nucleolus have reappeared in this telophase stage, and the two daughter nuclei are formed. You can see there are two daughter nuclei. One is this and one is this. Two daughter nuclei are formed. Okay. So what is happening exactly? Prophase, in prophase 1, the chromosomes are getting condensed. Okay, the chromosomes are getting condensed. In metaphase 1, the chromosomes are moving at the equatorial plane. Okay, this is the main punch point that you have to remember. Then anaphase 1, the chromosomes are moving away from each other, and at telophase 1, finally, these chromosomes are getting elongated, uncoiled, and the nuclear membrane and nucleolus reappears and two daughter nuclei are formed. You can see here clearly it is seen two daughter nuclei are formed. Okay, let us move further. After telophase 1, as we have studied in the mitosis, that there are four phases, that is prophase, metaphase, anaphase and telophase. And after telophase, the process of cytokinesis takes place. Same here in the process of meiosis 1 also, prophase 1, metaphase 1, anaphase 1, telophase 1, and then will come the cytokinesis 1. Okay, this comes after karyokinesis. As it comes in mitosis, it comes in meiosis also. After karyokinesis, cytokinesis occurs and two haploid cells are formed. In many cases, these daughter cells pass through a short resting phase or interphase. Let us study first in detail about the cytokinesis. Cytokinesis involves the formation of cleavage, that is furrow, resulting in the pocketing of the cell into two cells. You can see two cells are formed. At the end of the telophase 1 and cytokinesis, two daughter cells are produced, each with 1/2 of the number of chromosomes. Haploid set of replicated chromosomes. of the original parent cells. You can see from the parent cell, half of the chromosomes are here in one daughter cell and half of the chromosomes are here in the another daughter cells. Okay, so chromosomes are divided into two equal parts. That is, they are divided into half each. Okay, so after this, in many cases, daughter cells pass through a short resting phase or interphase or interkinases. Okay, it is also known as interkinases, also known as interphase 2. Okay, it is similar to interphase. That means in this, what is happening? The daughter cells pass through a short resting phase. Okay, in some cases, the changes of the telophase may not occur. Telophase, there may be no changes in the telophase. The anaphase directly leads to the prophase of meiosis 2. Okay, so this stage may occur or it may not occur. It is the resting phase of the cell. Okay, and many cases daughter cells pass through the short resting phase, and many cases the cells don't pass through the resting phase. This resting phase, interphase, interkinases, all are one and the same students. Okay, so here we have completed the process of cytokinases 1, and with this we have completed with meiosis 1. Totally we have completed with the meiosis 1 part of the chapter. Okay, we have studied mitosis, then we have started with meiosis 1. Meiosis 1, 4 stages, that is prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinases 1. So we completed all the phases of meiosis 1. So in our next lecture we will be studying meiosis 2 in detail. That is prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinases 2. Okay, and the significance of meiosis, and then we will study about the difference between mitosis and meiosis. Okay, so we have completed more than half of the chapter. That is, we have completed with mitosis, we completed with meiosis, and in that meiosis also we have completed meiosis 1 completely. So in our next lecture we will start with meiosis 2. Okay, so for today we will stop our lecture here. We will continue in our next lecture. Thank you, students.